This is the homepage of the Omics Playground platform. It's a platform dedicated to the analysis of transcriptomics, proteomics, and metabolomics data. And we also have currently a multi-omics analysis module in beta that is not going to be currently discussed in this presentation. So the first step I want to go through is how to upload a new data set in the platform. So as you can see from the home page, I already have a choice that allows me to upload a new data. So I'm going to click here, and now the platform takes me to the page where I can select the type of omics data that I want to input. As you can see, we support different types of transcriptomics data, as well as single-cell proteomics, and as I mentioned, metabolomics and multi-omics currently in beta. So for this example, I'm going to choose a proteomics data set. And then the next step is to select an organism. As you can see, we support both human, mouse, and rat extensively, but we have added support to more than 200 species, as well as a support for a custom organism, for example, a new bacterium that you just sequenced. So in this case, I'm going to select human, and then I'm going to click Start Upload. So for the platform, you need two compulsory input files. The first file will correspond to your abundance or read counts tables. So in this case, we are working with proteomics data, so it's going to be an abundance file. And I'm going to load it I'm going to load the example file. And here you get a preview of your input file. And you can see the structure is pretty much standard structure for most of your proteomics and transcriptomics data sets. You will have your protein or gene IDs in the first column. They can take, especially for human, mouse, and rat, various forms. In this case, you see they are AG and Cs IDs, could be uniprot IDs, could be ensemble IDs, RefSec, and so on. And then in each of the following columns, you will have your individual samples with the protein abundances or read counts. In this case, you can see that I loaded a pre-normalized data set, but our platform also supports raw abundances or raw read count files. An important piece of information that you get from this page is that you can access the documentation from our database. And this provides you with more information about the structure of the input files. And you can see here the different input files that we have in the platform, as well as some important checks that you need to make before uploading a data set. And in particular, you should pay attention to the characters that are not allowed in the different input files. So after you have inputted your abundance or read count table, you can go to the next step where you will upload your metadata, which here we call samples.csv. And again, I'm going to load the example just to show you that this is a simple file that you can even prepare with a spreadsheet program such as Office. And the structure will be in the first column, you will have your sample IDs, and of course they have to match the IDs that you used in the read count table or in the abundance file. And then in each of the following columns, you will have your different conditions. So in this case, for example, we have activation state, we have time points, and then we have the combination of activation state with time point. The next step consists of creating comparisons between the different groups in the different conditions. You can also upload that as an external file. However, if you have relatively simple comparisons, I suggest using our interface. And with our interface, it's very easy to just select a phenotype or even combine multiple phenotypes, such as in this case, I'm going to combine activation and time. And then it's a matter of simply moving your control, groups into either control group or main group. And you can do it either one group in each of the two boxes, as in this case. Then you can edit the name if you want to. In this case, I'm going to remove the prefix and just add it to the list. Or you can even combine multiple groups. So for example, here I'm going to take just the earlier time points versus the later time points. And now I'm going to edit this to be a bit more informative. So just late versus early. And one important thing to notice here, when I do edit the name, I always leave this component, so underscore versus underscore, right, intact, because this is required by the platform. So once you have finished doing your preparing your comparisons, you can go to the next step. And this is where you will have some quality checking as well as other aspects such as imputation of missing values, normalization steps, removal of outliers, and batch effect correction. Missing values applies generally only to proteomics data. And if you click on impute, it will give you a choice between three different approaches. Now you can see that in this case, I don't have any missing values in my data set. Chances are that you will have some in your data set. And you can also select the different conditions for retaining rows that contain missing values. So you can be quite stringent or generous, and the ones that are retained will then be imputed. You can also ask the platform to treat zeros as missing values. However, I suggest for novice users not to just apply these randomly. There is actually quite a few drawbacks that come with imputation of missing values. So I would suggest you carefully read about imputations before applying it. The next step is normalization. And in this case, as I mentioned, we already have pre-normalized data, so we could easily skip this step. However, if you have non-normalized data, so raw data sets, you will have a choice of different approaches that you can use to normalize the data. The default is to apply max median normalizations. We also have max sum, quantile, or you can choose a reference gene, or sometimes what people like to do is add spike-ins, so you could use that as a reference to normalize the data against. For transcriptomics data, you will see different options. So there will be, for example, CPM plus quantile normalization. So this is, again, very specific to the type of data you are analyzing. The removal of outliers is something that I do not recommend performing the first time you run a data set. You definitely want to check the presence of outliers visually before you apply the automatic detection of outliers. And ideally, you will find that there is usually a good correlation between the automatic detection and the visual analysis. And finally, we have batch correction. Now, batch correction is a bit of a double-edged sword in the sense that it can make your data cluster much better, but at the cost of adding potential false positives. As you can see in our platform, we have 5 different approaches that can be used to normalize the data. We have Combat and Lima, which are supervised approaches. And then we have NPM, RUV, and SVA, which are non-supervised approaches. So if I select remove batch effects, I can choose which approach to apply. And I would normally suggest trying first supervised approaches where you know the batch effect. So for example, if in your data you have batch effect that is correlated with day of sample collection or lab of origin, you can add that at the input stage in the metadata as a condition, and then you can choose it as a batch parameter. Otherwise, as you can see here, you can also perform combat and lemma using auto detection and let it cluster the data in a fashion kind of similar to unsupervised detection. With the unsupervised approaches, everything will be done automatically. So there is nothing that you need to specify here. But they will also tend to apply more drastic changes to the expression profiles that you get in your genes or the abundances that you get in your proteins. So again, I strongly recommend running the uncorrected data first, then applying batch correction and comparing the effects that batch correction is having on individual protein abundances or individual gene expressions, and make sure that the two are pretty well correlated, something like log-fold change correlation across the board of at least 0.95. Otherwise, you risk introducing a lot of false positives in your data. The final step is where you can choose computation options. As you can see, we have different options. The first one relates to feature filtering. And here we can, for example, apply different ways in which we deal with the data. So you can see that by default, we exclude features without symbols. So basically non-protein coding, or in this case, non-human protein coding entries. In general, if you're only interested in the species you're working with, or you're only interested in protein coding genes, you will leave this on. But if you're working, for example, with transcriptomics data and you want to include non-protein coding genes, such as pseudogenes or long non-coding RNAs, you will want to remove this option. The other options include ways in which the platform can deal with duplicated features, and also whether you want to append symbols to features IDs, especially if you are working with other ID types than A, G, and Cs. I normally would recommend leaving it as default, unless, as I mentioned, you have some good reasons, for example, to exclude all zero features, or whether you want to include also non-protein coding genes. The gene tests or protein tests will depend on the type of data you're working with. So in this case, I'm just getting a single test because I have a relatively low number of samples in one of my groups. Generally, you will see for proteomics data, trend lemma and t-tests. For transcriptomics data, you will also see dseq2 and edgeR, and you can run multiple tests at the same time. For the enrichment methods, again, we support different algorithms. The default choice is to show Fisher, FGSEA, and GSBA. SSGSEA is also a pretty good option, but tend to be also more time-consuming, especially for larger data sets. And rank correlation is also quite popular for proteomics data analysis. The extra analysis is normally something that is present by default, and I would run for all human, mouse, and in fact, generally vertebrate data sets. They include supports for cell type deconvolution if you're working with single cell data, but this can also be useful for bulk data in case you want to look at, for example, contamination across your sample groups. There is support for the drug connectivity and drug sensitivity databases. We also perform experimental similarity analysis. I think this is useful basically for any species you're working on, because it allows you to compare across all the data sets that you uploaded into the platform. And we also have support for gene network analysis in the shape of WGCNA. Finally, for more advanced users, you can also add a GMT file into the platform that will include gene sets that are currently not present in the platform. So there is an example that you can extract from the platform. So if you want to create your own GMT file, it will show you exactly the structure it has to take. And generally, I find this particularly useful if you're working with non-canonical species, where you might have specific gene set collection that obviously will not be supported in the platform. So for example, if you're working with some parasite species and you want to include KEGG files, KEGG IDs, or some KEGG pathways, then you will want to add it as an extra GMT file. So once you have selected all these options, you can click compute, and the platform will take anything between 5 minutes to maybe 2 hours if you're working with single cell data to compute all the analysis. And then once you're done, it will take you to the home page where you will see all your data sets, and you will be able to select then one for the analysis. And loading a data set in memory is very simple. All you have to do is highlight the data set and then just click load selected. And in a couple of seconds, it will load it into the platform ready for your analysis.